Okay, thank you very much. Welcome to this presentation. Eleva, as you said, is not very well known because we were flying under the radar for a very, very long time. The company actually exists since more than 20 years. It raised more than 140 million euro venture capital so far. We have currently about 50 employees. I don't go to all these points. We have a special technology platform, which I will explain in a minute. And we have 2 clinical assets about which I also will share a little bit of insight. So the thing is, it's a production platform for recombinant proteins, but based on MOS. Basically, it's the same as the CHO process. Everything is the same, only a different organism. For those who know CHO production, you know that there can be pain sometimes. CHO are very sensitive, especially with temperature, with pH. They don't like upscaling very much. This is plant-based. The plant cell has a cell wall. It's a very robust system. Upscaling is no problem. From when it grows in 10 liter, it will grow in 500. You can go this step immediately. We have shown that we can express proteins that don't work in CHO at all. CHO doesn't like very long, complicatedly folded proteins. We can do that. One of our clinical assets is one example like that. I will talk about this later. And another thing is cost of goods is much cheaper with us because we have a very less complex growth medium. And the cost of goods in average are about 20 to 30% less than CHO. We work with this plant, with the MOS, but only you can't see it here. I have to walk, sorry, camera. So you have in the life cycle of that plant, you have a part where it's single cell. They grow in filaments, but it's single cell, and that's what we are using. So you basically grow single cells in solution like you do with CHO, no difference. Here are a couple of pictures for that. The advantage is that because at that stage in the life cycle of the plant, it's haploid, so it's easy to manipulate on the genetic level. Although on the other hand, it's evolutionary seen. It's a very modern plant, so to speak. It has all the features for post-translational modifications that you want to see in a protein for therapy. It grows in simple medium, I said that already, and it is, the last point is not so important. So if you compare to the other systems, especially also to CHO, which is the workhorse in the industry and will stay that for a very long time still, but if you compare it, you will see that we have a couple of advantages in all of those I have mentioned already. So we believe that our platform will not replace CHO. That would be a little bit too high to reach. But what we can say is if you have a protein that is difficult to make in CHO, try it in our system, probably we can salvage your project, right? So this is just to illustrate that the whole process is the same like with CHO. From the beginning, when we started to develop that, we made sure that every step is the same as in CHO. The only difference is really that it's a different organism growing in the same, in our case, 500 liter fermentation, single-use bags. The bag holder is Sartorius, all the same, all right? Here, that's some pictures from our GMP suite. You can see it's if you switch off the light, you can see the green glow. It's very pretty. The important thing is we have in the moment it growing in 500 liters, and we have 2,000 liter GMP. We are just now growing this up. Our goal is to have in 2 years about 6,000 liter GMP capacity. Maybe in this context, one word. We are the technology is accepted by the authorities. We have 2 assets in the clinic, so they all said yes, it's fine, no safety issues whatsoever. They liked it because it's even in their eyes even more safe than CHO because you don't have any animal-derived additions in your growth medium, for example. The other thing is that also the industry is now accepting us. We will, in a couple of weeks, we will announce 2 strategic cooperations with large CDMOs who want to help us to increase our capacity and who want to use it by themselves as well. and are buying licenses from our technology. So stay tuned for these news. Now to the assets. The one is alpha-galactosidase for Fabry disease. I go through this very quickly. There are two assets. It's a rare disease. There are two assets on the market, two projects, one from Sanofi and one from Replagal. And we have head-to-head comparison to both, and we can show that we are probably better in the patients. So one point here as an example, you can see that Replagal, that's the Takeda product, which is on the market, is very unevenly glycosylated, and it's especially mannose-6-phosphate. Doesn't matter. Our glycosylation is more evenly distributed, and anyway, from our system, our glycosylation pattern is always much closer to human than CHO. But this is, in this case, very important because we have mannose on the surface instead of mannose-6-phosphate. What happens is then it goes, the enzyme then goes through a different receptor into the cell, into the target cell. It goes there into the endosome, which serves as a kind of intracellular depot. What happens is, and this is then depicted on the next, what happens is it stays longer in the cell. You have a faster uptake into the cell. This is Replagal on the market. This is ours. You have a faster uptake and a much longer distribution from the cell again. And these are data from our phase 1. In patients, you can see then that even after 3 weeks here, the marker doesn't go up anymore, which shows that we are still active here after 3 weeks, whereas the two that are on the market from Takeda and Sanofi, they have to be infused every 2 weeks because then they lose already their activity. So we can show, at least at that point, very easily that we are better than the two on the market. We, in the moment, assume that we will have, instead of every 2 weeks infusion, we will have every 6 weeks infusion, but we need more data for that. All right. Second asset is factor H, also enzyme replacement therapy, in this case for complement disorders. Complement disorders is a family of 8 diseases, all rare diseases except of one. The one that is not rare disease is dry AMD, which leads to blindness. The other ones are very much smaller diseases. I will tell about that a couple of sentences soon. An interesting story. In the late 80s, early 1990s, there were a couple of clinicians. They were able to isolate factor H, which is the protein we are talking about. Were able to take that out of serum samples. It was very laborious, very cost inefficient, but they were able to treat 5 patients with that. So in these patients, factor H is not there or not functioning. The immune system is overactive. It's not switched off, so to speak. These patients get very sick because of that. So these 5 patients were treated with serum-derived human factor H, and the results were really spectacular. So after that, many people tried to produce it recombinantly in all systems that you can imagine. It didn't work at all. What's the time? And we are able to do it in our system, to make it short. And we are going into the clinic. And sorry, this is such a stupid slide. But what I wanted to say here with that last slide is just that the competitors, which are now under development and 2 are on the market, they are all inhibitors. That means they switch the immune system off at that time, at that stage, which works, but the patients get very sick and open for infectious disease. We give back the regulating molecule factor H, the human identical, human glycosylated factor H. And this is just a regulator. So the immune system is going back to normal and you avoid all these side effects. Okay, we will have first patient in Q1 next year. Sorry for the time. It's a long story. We have head-to-head comparison to a lot of these assets. Like here, for example, we can show eculizumab is one of the inhibitors that we protect still the patient from, in this case, a Neisseria infection, which eculizumab, which is on the market, cannot. Then this is just the family of indications you have here. It's a very, very, very big market, especially also dry AMD. We go into the clinic now in C3 Glomerulopathy, and but we have preclinical data, including animals, to all other indications. and can show that it works. Then for the investors amongst you, we just start now a pre-exit mezzanine round. So heartily welcome. We are. The goal is to have a consortium together until early next year, so that the money comes to our bank account summer next year. That would be lovely. Quickly, the team, that's our C-level team, and the disclaimer, and that's it. So if you want to discuss, I'm already 1.5 minutes over time, so thank you very much. But if you want to discuss, please find me outside today or tomorrow. Thank you very much.
Thank you.